It's 4 o'clock, and it's time for our guest seminar speaker. So today I have the pleasure of introducing Mr. Justin Stevens. Before we begin, are there any announcements from either students or faculty? No? Great. So Justin serves as the Sea Run Fish Ecosystem Project Coordinator for Maine Sea Grant. He has over 18 years experience with Sea Run Fish in Maine and has previously worked for the National Oceanographic and Atmospheric Administration's Northeast Fisheries Center and the Maine Department of Marine Resources on Atlantic salmon and other sea run fish. His research spans freshwater habitat and population assessments for Atlantic salmon and has a more recent focus to the Penobscot River estuary. He has led a survey on the estuary using hydroacoustics to monitor changes in fish distribution during the period of large-scale river restoration since 2012. His interests are also in the ecology of sea-run fish, including Atlantic salmon, rainbow smelt, alewife, blueback herring, and they all utilize the estuary in different ways during their life cycles. He collaborates with various researchers to explore and what might be drivers of the growth, survival, and distribution of sea-run fish for the estuary. Justin received his bachelor's degree in science in biology from the University of Maine in 2001 and received a master's in marine biology also from the University of Maine in 2019. So thank you so much for coming all the way to Castine to let us know more about your work.
All right, well, thanks everyone for joining us today, and I really do appreciate the invitation. I was telling Stephen I haven't been down to Castine yet in 20 years of my career or with recreation, so really happy to come down here today. So I'm going to start out just giving a little bit of background and history on sea-run fish and the types of things that we're investigating. I've got a lot of different research as the introduction suggested that I work on. So I was just going to try to show you kind of the highlights, things that we've got going on at Sea Grant right now. And then in context of the Penobscot River and its restoration, a lot of the research is focused on that. So I'm going to spend a particular amount of time on that. And I was also asked to talk about career path, and I'll call it kind of my lessons learned getting to this podium today from the University of Maine over 20 years ago. So I can understand that we're going to leave ample time for questions at the end, so I'll make sure I'll keep an eye on the clock and not take too much time. So just a little bit of background and history. My project and all my work revolves around sea-run fish, diadromous fish, and they all share a common life history in that some of their growth is in freshwater and some of their growth is in the ocean environment. And what's fundamental is the connection of those two environments to complete their life cycle. So here's a depiction of Atlantic salmon. I've worked a lot with Atlantic salmon. I think they're pretty interesting. And just coming down into the hallway, I realized a unique connection with the Maritime Academy. So here we are in Maine. A salmon will spend a couple years growing up in freshwater habitat. It'll turn into this migratory phase, the smolt, and then it swims up into the Labrador Sea to Greenland. And for example, we just had part of an international collaboration. I think it was the 52nd year that there were scientists at ports along Greenland that were sampling Atlantic salmon and trying to determine how many salmon had come from Maine and other rivers here in the Northwest Atlantic. So salmon aren't the only species, though. Here in Maine, we have 12 diadromous species, or like I said, these fish that rely on the interface between the salt and the fresh water. They vary from the silvery-looking herrings to the sturgeon species, range in size from the smallest, I guess I'd call the rainbow smelt, 6 or 8 inches long, to Atlantic sturgeon that are passing by Castine here that could be over 8 to 10 feet. So a lot of diversity within this sea-run fish collection. So as I mentioned, fundamental to their biology is this connection. And if we go back in time a couple hundred years, we had large populations of sea-run fish in the Penobscot River and other coastal rivers in Maine. But what happened during the Industrial Revolution were that dams were built, and when they were built, they didn't provide much for fish passage. Because of the abundance of the fishery, of the fish, there were also robust fisheries, so this was a common sight, anglers taking advantage of the abundance. And the combination of these two, habitat degradation and the dams and overfishing, really resulted in the loss of all of these fish. And most importantly, their ecosystem function. So I'm going to get into that in a minute, but we can already think about what type of function a fish could have that's grown from 2 or 3, maybe 4 inches long in fresh water, gone out to the ocean and grown 8, 10 times that size and come back into that freshwater habitat to spawn. So I like history a lot. I like fish biology, but I like history too. So I think this is an interesting quote from 1873, and the words are here, you can read, but the same sentiment is true today, where people are making the connections that the loss of sea-run fish have impacts well beyond just that species and just those fish being lost. Here the commissioner cites the cod that have disappeared and the fact that this is happening at the same time as these loss of fish in inland waters. So we've known for a long time that there's this connection, but it took until really the early 2000s that the scientific community was starting to formulate some of these hypotheses and investigate the mechanisms here on the East Coast. And along this time, there's been a lot of changes. Nowadays, there's a lot of changes, including instead of building dams, we're taking out dams, and instead of overfishing, we're stocking. And what it's really doing is restoring the populations of sea-run fish as well as their habitat. And again, I'll say it a number of times today, restoring these connections between the freshwater and the marine environment. So this is a picture from 2013. This was the dam that was up near Bangor. They put excavators right in the river to remove it. If you go here now, I was there just a couple weeks ago. Where this stood, there's just trees that are starting to grow this high, and instead of concrete stopping the flow of water, it's just a continuous riffle of water. This one was just upstream, Great Works Dam. This was removed in 2012. Same thing. It just looks like a pretty natural river now. And what that's done is in this lower right picture, this is at the next upstream dam where they installed an advanced fish passage system. And we've got sites like this where you've got several species of sea-run fish coming back to spawn in the Penobscot River. That's an Atlantic salmon, alewife, and a sea lamprey. So I should have put this map a little bit earlier just to orient everyone, but a lot of the work, like I said, I've done has been in the Penobscot River. This is the area of Milford Dam, what I just mentioned had that viewing window. This is where VZ Dam was that was removed. And then this is what I call the estuary, roughly from Bangor down to Fort Point. And then you can almost see it, that biomass or fish biomass was a key metric in understanding these ecological interactions. And because everything that was going to go to freshwater had to go through the estuary, that this interaction was likely to be occurring down in the estuary. So here's a timeline. I'm going to focus mostly on the post-restoration activities, but we did a lot of work in the early 2010 through 2012. Down here in this box, finding beach seine sites and setting nets and all these different things. the estuary is a really difficult place to do fishery sampling because of the tides and the currents. So we didn't have as much luck with all of this gear, but we did a lot better with the hydroacoustics. So that's what I'm going to talk more in depth on. And we spent those first 3 years and then developed a report and recommendations on what would be the best path forward in monitoring the estuary. And as I just mentioned, the acoustics was going to be the gear of choice. But the other part that was kind of leads into my next part of the talk, some of the surprises that we found, that the estuary was not a desert. Like a lot of people we had talked to, the freshwater community was really well studied. We knew what was going on. A lot of people in the ocean were doing fisheries work, so we had an idea of what the fish that were out there. But how and when and what used the estuary in terms of fish were really, it was really a data gap. So we found it was really complex. There was a lot of fish. They were there most of the time that we were there. And more importantly, we found that most all the diadromous species were already present before the dams were removed. So we had the building blocks of restoration even before there were any management actions. So the next few slides, I'm just going to highlight some of the surprises. This is kind of in that lessons learned too of the career path, but we get into a project and you start somewhere and there's usually, a hypothesis that you're testing, if it's a well-designed study. And our hypothesis was that we were going to look at the changes in biomass over time as restoration proceeded. And pretty much the first day we started, we were just in for a surprise. It just seemed like every time we went out, we found something that we didn't expect. So this is a good example. American shad. They were known to be in the river. Historically, folks thought that they were either extirpated or there were so few that we would never be able to detect them. And the first summer we were out, we found not only that they were there, but they were reproducing. So we found the little juveniles in these beach seine halls. So we proved that, that they were there and spawning. The other paradigm that we had that we were working under were the river herring. Their life history were that adults came up in the spring, they spawned in fresh water, they went back out to the ocean, and the juveniles stayed in fresh water for a couple months, ran out through the estuary and lived in the ocean until they spawned. Well, every time we went in the estuary, we found these juvenile river herring, suggesting that they were using it as a long-term nursery. The other thing that we found were that this wasn't just going to be about measuring the fish that were coming in and out, that we'd have fish like Atlantic herring. We're using it as a nursery. We had the juvenile river herring. We had smelt that were coming in, as well as the adults. So instead of just, a community that had members that were coming in and out at pretty predictable time periods, we saw that it was much, much more complex. So as I mentioned, we always would find these juvenile river herring in the river. I was really lucky to get into a collaboration with folks at the University of Maine that were interested in chemistry. Specifically, what we did was we took the otoliths, these little bones in the fish, and we took a cross-section and looked at the microchemistry along a transect from the origin of the fish towards its birth to when we captured it. And these were alewives, so this is typically 4 or 5 years. And then what we did is, based on other literature, we picked out elements that we thought might be able to provide a history of where that fish was in terms of relative salinity habitats. So I know there's a lot on this graph, but this is actually raw data from a laser ablation technique that provides this element. And it just goes along this transect. There's this big change, and then it comes along this new baseline. So this is what we expected to see when we looked at alewive otoliths, because we knew that they were born in fresh water. So we predicted they would have low strontium, and then we predicted they were going to transition through the estuary and into the marine habitat, and then that they were going to live out the rest of their life in marine habitat. This just shows it a little bit clearer that we thought there would be this definite, shift between habitat types. And we did find that in like 12% of the alewives that we looked at. So it was good that we did find that habitat type, or the habitat usage. But what we found more often were that it was much more complex. So we saw these fish that had these multiple migrations between freshwater and the marine interface. So this helped us understand a little bit better of why we were seeing juveniles in the estuary for so long, because some of them had probably come down from freshwater, gone out to the ocean, but then came back into the estuary. And there might have been some that were going back and forth multiple times. And it ended up being a pretty high proportion. I'd say over a third of the fish that we analyzed had this life history. And other folks have called this a non-textbook life history. But I do a whole talk on that, and the whole end of it, as I said, we probably shouldn't call it textbook because these fish don't seem to really follow any textbooks. All right, I'm doing good on time. So the next part, I just really want to focus in on some of this long-term monitoring that we've done and the restoration response after the dam removals. So as I mentioned, there's been counting that was going on at the upstream dam in Milford. And this is a graph of the counts of river herring. So there's two species that look a lot alike. This is an alewive, and this is a blueback. And they started counting in 2010, way before any of the dams came out. And so you'll notice the scale, we basically had thousands of river herring to start with. And in 2014, this was the first year that they started counting fish after the dams came out. And you'll see the increases. And you could just add on a couple more years here. I didn't have room, but 21 and 22 are basically around this same level. So the river is counting around, producing around 2.5 million river herring now, as compared to a few thousand. So if we think about the monitoring in the estuary, this is one of these points where we know we have a count on what's happening. Those adults are coming up through the estuary and going to Milford, spawning up above Milford, and then their juveniles are coming back down into the estuary. So we know, because of these counts at the dam, there's been a change. It's gone from 1,000 just adults to 2.5 million. So as I mentioned, this acoustic survey has been going throughout that entire time series as well. I'm not going to talk a lot about the trawling that we did, but that was basically a ground truth method that we used to validate the hydroacoustics. And just so we're all on the same page, and I understand a lot of folks are working on different things, so if this is a review, I'm sorry, but if it's not, then it's good to get everyone on the same page. When I talk about hydroacoustics, we basically have a really expensive fish finder. So it's a sonar that is calibrated to specific frequencies, and what we're taking advantage of is the fact that fish have an air bladder. Fish that don't have an air bladder, we don't pick up as well with the hydroacoustics, but all of those species that I have been talking about, the herrings, the salmon, and those, they all have an air bladder. So what this allows us to do is to tell the relative density based on the signal that we get back, as well as the relative size. A lot of what I do is this data processing. I luckily still get out in the field like 20 times a year and get to do the surveys, but there's at least that amount of time that I'm in the office looking at the data with a view like this. So we go through and we've got to process the hydroacoustic data. We'll also take advantage of programs, computer programs, there's algorithms that predict what targets are coming back as fish and approximately what size. So we can produce some size distributions like this and convert it from the decibels, which is an acoustic unit, to biological units like centimeters. So the other part that we can do is to take some assumptions and jump from the raw acoustic data, look at some target strength distributions, like I said, these size distributions. We developed our own size to length regression and also a length to weight regression so that we could get the holy grail, which is fish biomass. Because when you talk about this, and I'm a fish biologist, I said that in the beginning, I'm not necessarily an electronics engineer. When you're talking about decibels and all these other things in a scientific audience, there's only a small subset that's going to understand that. If you talk about how many fish, how many metric ton of fish, that's a unit that more people understand. So we worked hard to try to get all of our data down to these more meaningful units in biomass. So all that to say, all that work, it was like, well, it was my master's thesis, and then about 5 years of work before that got this graph. So these are all the surveys that we've done since 2012 through 2020, and this is the total fish biomass in metric tons. It's messy because there's seasonal variation as well as annual variation. But in general, what we found is that the fish biomass in the post-restoration years, since the dams have come out, is generally higher. So where we had surveys, we were getting around 50 metric ton per survey. Our highs are now in the hundreds metric tons. And then this is just another way to display that information, because I mentioned there's seasonal variation as well as annual variation. So in general, there aren't a lot of fish early in the spring. There's more fish as they come in and migrate. Fish come in from the ocean that are like seasonal residents. And in the fall, a lot of fish go back to the ocean and aren't in the estuary. So you see this kind of seasonal biomass trend. But when you plot it like this, you see that even within the spring surveys, the spring biomass in the early years, 2012, 13 and 14, is lower than in the post-restoration years. And similarly in the summer, and then here in the fall. What causes a lot of that noise in that previous graph is that sometimes the fall biomass isn't usually as high as the summer biomass. And then this is just another taking that biomass calculation one step backwards, and I just was going to talk about the fact that it's fish size and density that contribute to biomass, and some of the work that we did shows that what's really driving the overall increases in biomass is the fish density. So the fish size hasn't really changed that much, but what's changed, and this graph looks very similar to the previous one, because fish density is really driving those changes in biomass. So yeah, and this is in acoustic units, but the point's the same, is that the early years where fish hadn't been restored are generally lower in each season. So I skipped over it in the methods slide that I had on the acoustic survey. But another interesting thing that we have done when we're doing the hydroacoustic surveys is counting birds and mammals that we view along the transect. So we start here at Fort Point, and we're coming up through, and we go by several known seal haulouts, as well as at least one popular cormorant rookery. So this was definitely not the main focus of the research that we did, but it actually provided opportunities for advancing the project with partnerships and folks that we hadn't been working with before to look at distribution of these other species. So for example, cormorants, that's these blackbirds here. They're migratory as well as the fish. So in the spring, they come in from southern states, come up here and breed, and then they pretty much all leave in the fall. And But if you track the number of cormorants that we see per survey, there's generally an increase over the same period that we've seen the increase in the estuary biomass. Again, with all the same noise that I just talked about because of their seasonal migrations. So one thing that was interesting that we looked at when we looked at seal abundance is that seals really didn't increase in their abundance over the time in this estuary. But the other metric that we had was the number of salmon that get handled and whether or not they have a seal bite. So some of the salmon, when they come back from the ocean, if you're handling them in that trap, they literally have canine punctures on their fin or on their back. And we don't have a lot of species that have canines except for seals, so they're almost all called seal bites. So we worked with a researcher that was, an expert in marine mammals, and they looked at all the data. And even though we'd seen more sea-run fish and we didn't see any more seals, there was a significant increase, a decrease in the amount of salmon that have been observed with these seal bites or these seal injuries. So we explored in this paper that could be a couple different things that are going on, but one of the leading hypotheses is that maybe with more forage type fish like herring and the alewives, that the seals are going after those more abundant species versus the Atlantic salmon, which are still relatively in low abundance. I should have mentioned that. The population has pretty much stayed still at around 1,000 individuals a year, which is, again, way below the 2.5 million of just the river herring. So I know that was a ton of research and findings in the science, but I've summarized it down here that we're seeing higher biomass and density of fish in the estuary at the same time that we've seen these population increases upriver. It's a complex system. It's a mix of diadromous and these marine species, but we're also starting to investigate some of these predator-prey interactions and getting a better understanding of some of these hypotheses that folks had proposed. And then finally, we still don't know all of the details, and the idea is that we're going to continue this long-term monitoring as long as we can to better understand the changes. So, yeah, I think I've successfully done it. We've got to a point where we can talk a little bit about career paths. So, in the intro, mentioned that I started with an undergraduate degree in biology at the University of Maine. I spent about 9 years with the Department of Marine Resources as a biologist with them. And then I moved to NOAA Fisheries for another 8 or 9 years. While I was at NOAA, I went back to school and got a master's degree in marine biology. And when it was right about the time that I got that degree that I came here to Maine Sea Grant. So I wouldn't say that I have a conventional path in my career. A lot of folks go right from their undergraduate to their master's degree and their PhD. I didn't do that. I wanted to get out in the field. I knew that the fit at the school that I was looking at wasn't there for me yet. I just didn't want to make that leap, so I went to work instead. But what I had done while I was at the University of Maine was I took advantage of internship opportunities. I started to meet professors that were doing work in fisheries, what I knew I was going to be interested in a career. And it was thanks to those relationships that I got the first job that I did. So I was on a doctoral candidate's field crew as an undergraduate, and she ended up working for the agency that I was hired on. So I built those connections early on in my education. I came away with a all the great skills that like you're learning here. But when I went to work for DMR, I realized how many hands-on skills that I hadn't acquired yet. And I was thrown right into it. I'd never whitewater canoed at all, and we had to stock 2.5 million Atlantic salmon fry out of these coolers, like the first month I was there. So, and there's no course that you can take to learn that. Like, you just literally have to go out there and do it. So, I was really lucky. I had great mentors and learned a lot about, you know, navigation and fish handling and all of these different things. So it was also what I learned early on was just how great of a career that fishery science, or just the science in general, is, because you're immersed in your subject. This was during a habitat survey. We were out looking for different types of habitat, and you're interacting with just all of the beauty of that environment. So I learned different fish capture techniques like electrofishing, which when they first told me what we were doing, I'm like, what, we're wearing electric shockers in the water while there's electricity, and then we're supposed to catch them? It just didn't seem like that it was a really good idea. But yeah, after a couple of seasons, it's just second nature. You just stand in the water with electricity, you know? We also got to see some really cool things. As I mentioned, Atlantic salmon aren't very abundant in the state of Maine, but we could go out in the fall. That was one of my favorite things to do this time of year as a salmon biologist. Go out and count the nests that the female salmon were digging. So this is a big area of disturbed gravel. If you canoe in a reach where there's a lot of salmon digging these reds, they just kind of look like light circles in the dark gravel. So then when I went to NOAA, I brought all of those biology and field skills that I had, and NOAA Fisheries was particularly focused in on age and growth questions for Atlantic salmon. So again, I never really learned this in any of the coursework that I had, that you had the ability to age fish with their scales. And NOAA Fisheries in Maine, they're involved more with that migratory phase of the salmon, so smolts. So this was another thing that I learned how to do. We run these rotary traps, so the salmon come down the stream, they're headed to the ocean, which would be over here. They get intercepted in these traps. Then you've got to canoe out to them, take the salmon out, measure them, do all kinds of interesting things, take genetic samples, and then let them go. But when I was at NOAA Fisheries, I was also working on this estuary project, and we were doing a lot of collaborations with professors at different institutions in Maine, and it was through that I got to know a couple professors, and they said, well, you know, you're always bringing out graduate students and doing all these work on the hydroacoustic survey. You should get a master's degree. And I said, well, it's kind of late. Like, I'm already in my 30s, and I don't think I can do it. But they encouraged me, and I did it. So I was super happy to get back. And I don't know if I'd necessarily recommend waiting that long to get your master's, but I appreciated that I had some experience when I went back to school, and I sat in a lecture, and I could interact in a way that, you know, I could bring my experience, but then, you know, also apply that to the coursework and things that I was working on. So I think the other really important component of professional development is networking. I mentioned that one of the first jobs that I had came from a direct result of working with a graduate student at the university, and really, I show this is my colleague, Dan McCaw. Dan is the fisheries biologist for the Penobscot Nation now, and he has been almost a decade. In this picture, Dan and I were both just seasonals that came out of our first job into the Department of Marine Resources, and we've stayed close the whole time. And we work on projects together, we go for funding together, we collaborate together, and it's these networks and collaborations that you develop during your whole career. You never really know, how they're going to transpire. So this is just a picture of, I think, the last proposal that I worked on, the organizations that were involved in some way, shape, or form. And it isn't like who that is necessarily that important, it's just how many. So most of the work that happens now, whether you're at a university or if you're at a state or federal agency, occurs with a collaboration with either other state and federal agencies or nonprofits. And in Sea Run Fish, there's a lot of different nonprofits. The Atlantic Salmon Federation is an international partnership. The Nature Conservancy is worldwide. There's really local partnerships like this Project Share that just works in down east and coastal Maine. And as I mentioned, the state and federal partners and the tribal partners. So, and as I mentioned too, Dan is a good example, but what I've seen is now as I work with more students that the undergraduates that are coming together as a cohort in a program, they often branch out. You're not all going to work at the same place, but they maintain connections professionally after that academic experience. So this is just a picture of all the interns that I've worked with in the last few years, and they've come from the West Coast and the Southeast, the Southwest United States, and have worked all over, but they all have this common connection, that for at least some time that they worked in Maine. So I just wanted to transition, and I struggled with where I lead with this or I put it at the end, but anyways, it's at the end. I just thought it'd be good to tell everyone about Maine Sea Grant, what Maine, that's where I'm working now, just what types of things that Maine Sea Grant does, and then I'll mention a couple opportunities at the end. So this is our motto, Marine Science for Maine People, and this is Sea Grant's mission. It's really broad, and it's intentionally so, because we really, it's hard to box in all of the different things that we work on. It can be anything from, for example, Sea Run Fish. I could be working with partners that are restoring habitat 100 miles from the ocean, maybe replacing a culvert or taking out a dam. And it's a nationwide network. So these dots are all of the Sea Grant programs. There's 34 around the country, and you'll see that they're surrounding all the Great Lakes as well. So once you're kind of in the Sea Grant network, like working here in Maine, you could very quickly see yourself partnering on a project with someone at Oregon Sea Grant or the Southern California Sea Grant. That's a great example. They have a salmon program, a position that's just like mine. They study a completely different species, but a very similar suite of issues that are involved in Southern California, which is just about as far as you can get across the country from each other. But I think what people get confused about too is that, is Sea Grant like a research facility? Is it a communications? Is it education? And I think people get confused because the answer is yes, it's all of those things. So there's really three pillars this three-part approach to what Sea Grant mission really is. It's definitely to support research. So, I conduct research with Sea Grant, for Sea Grant. We also administer research calls where we provide money to, folks at other institutions to fund research. But then we also perform this extension service, which I've been there for a little over 3 years now, and it's really hard for me to find exactly what extension is, but you can think of it as being an intermediate between the research and the information that's being collected and the individuals that are using that information to make some kind of decision. So tomorrow, I might work with someone that is a select board at a town that's thinking about fish passage issues or restoring sea-run fish. The day after that, it might be bringing information to a regulatory agency that is working on some sort of policy. Legislators, there's just tons of people, basically as many people as you can think of that use scientific information. That's the breadth of the stakeholders that we perform this extension service of. So, and then the other big part of it is education. So training future scientists or just stewards of the environment. So we have a K-12 curriculum coordinator that works with schools in bringing all the breadth of work that Sea Grant does. I think I mentioned this, but just so that everyone's clear, like sea-run fish is a really small part of everything that Sea Grant does. We work on lobsters, aquaculture, coastal resiliency, climate change. There's just a ton of marine issues where there's this need of information and extending that information and getting it in the hands of decision makers. So pretty much anything you can think of, we probably have some involvement. Offshore wind is another one. So yeah, so I think this is my last slide, and I didn't know how else to transition to it, but so part of my job too is to administer an undergraduate intern program, and this is giving everyone here the heads up that we're going to be recruiting for positions for the summer of 2023. I would love to see some folks from Maine Maritime Academy apply. We update our website when those positions get finalized, and I'm going to make sure that I circulate those to the faculty here to make sure that everyone knows, when they're open. And more importantly, what we're really trying to do with this internship is pair up students that have an interest in the research that's going to be conducted next summer. So I'm actually not even sure exactly what the research projects are going to be because I'm working with mentors to develop those right now. But when we know those, we put those position descriptions right on the application. So you're going to be applying to a job description. And one of them might be helping the folks at the Milford Trap, count salmon and alewives, and then do age studies or something as part of that. So they're really specific jobs. They are paid and they last most of the summer. So I think this year they're going to be 12-week positions. So this has been actually a pretty big part of my job is to really try to get these students connected with mentors. So that is my email address. I should have shared the Sea Grant webpage too that's on there, but it's just Maine Sea Grant. If you search for that, it'll come up and you can see all the different things that we're up to. I think that's it. I'll take some questions. I think we had a question way in the back there.
Hello. So, you guys been observing the fish as they move through the river over the years and how they have changed with the dam removals and everything. Do you see or expect to see maybe a change in population or behavior as climate change and water warming happens?
Yeah, that's a really good question. Do I have to repeat the question, or did everyone hear it online and stuff? Okay. Yeah, part of the initial drivers of starting a long-term monitoring were just for what you described, because we can have population changes from dam removals, we can have targeted stockings and those kind of things, but we realize that all of that is occurring under this sometimes not so long-term changes in the climate. And there's been a number of folks that have worked on studies just kind of evaluating the vulnerability of sea-run fish to climate change. And most of the sea-run fish species, if not all, are highly vulnerable, mainly because of that interface. They rely on fresh water and the marine environment. When you make that life history choice, you also are assuming that the habitat that you're going to is ready for you. So if it gets really warm in the river, and then you go out to the ocean and it's really cold, all of those physiological changes that you're trying to adapt to are temperature driven. So temperature is a huge component of that, and you're exactly right. Climate change could affect how those interactions work and then the population. So, yep.
So, how effective are the fish passageways that are built into the dams, compared to just removing the dam itself?
Yeah. So, do you mean at that Milford Dam? Yeah, that's a good question. So, there are a lot of studies that are more species specific to help answer those questions. It's actually a pretty complex regulatory mechanism with the Endangered Species Act. Some of the species are endangered, like Atlantic salmon. So then studies are ordered by the federal government to evaluate how effective the passage is. So for Atlantic salmon, we have a pretty good idea. And the answer is that I think in the last 4 or 5 years, more than 95% of the adult Atlantic salmon have made it above that Milford Dam. On the way out, the Atlantic salmon smolts, the survival has been pretty high, like over, I think, 98%. So, but in theory, the percentage that would make it over just the natural river would be pretty close to 100%. So that's actually a component of other work that's being done, kind of modeling what those subtle differences might result in a population, say 10 years from now or 20 years from now, because I can just tell you from the work that I've done, like those little increments add up, because Milford is the first dam that Atlantic salmon would go above. There could be 3 or 4 more. So if at every dam they lose 2% of the population, then it isn't just a few fish by the time you get to the most upstream one. And same as the way down. So if almost all the fish make it below Milford as smolts, those smolts already had to go through 5 or 6 other dams before they got down to Milford, and they could have already occurred losses. They could have died already. They can't die twice. So that's part of that evaluation metric. But does that answer your question? All right.
One more question.
So I know you said you took genetic samples of the salmons that were caught in the little traps in the rivers that go out to the ocean. Did you happen to did you take a sample to test to see if there were heavy metals that were kind of corroded from the dams into the water, or see how that changes their behavior or anything?
Yeah, that's a really good question. I haven't been directly involved in those studies, but there are folks that are looking at different contaminants. Mercury in the Penobscot is a big one that folks are looking at, and now increasingly things like PFAS. So it's interesting. A lot of that work that's happening right now is being driven more on the water quality side, and what would be actually a really interesting talk down here, Steve, would be the Penobscot Nation Water Quality Lab. They're doing a lot of work on contaminants in the river now because the Penobscot people have relied on these sea-run fish as sustenance for thousands of years, and knowing whether or not they have that ability to safely eat the fish now is a real question that they have. It's an important question that needs to be answered, and actually that research is taking place right now to help answer that. So I don't have a definitive yes or no of what all the results are, but people are looking into it because it's really important.
Well, thank you again very much.
All right, thank you.